Hello, my name is Brad Yoder. I'm a professor and chair in the Department of Cell Development and Integrative Biology in the Hersing School of Medicine at the University of Alabama at Birmingham. I would like to thank you for your interest in our research program exploring new roles for cilia in mammalian development and their association with disease and birth defects. Cilia come in two primary forms. The first on the left are called immotile cilia, also called primary cilium, are found one per cell in almost every cell in the mammalian body. In contrast, on the right, we have examples of motile cilia. These would be found on tracheoepithelia, ventricles in the brain. They beat with a uniform wave that causes fluid flow or mucus movement across the surface of the cell. When we first started these studies nearly 20 years ago, cilia were thought to have little importance to human health. That view has dramatically changed, in large part due to the identification of variants in patients with complex syndromes that are now referred to as ciliopathies. We now know that cilia dysfunction impacts nearly all tissue and organ systems of the body, and that cilia function is actually essential for normal development. But in only a few cases do we currently understand the role that cilia play in these tissues and how they lead to these phenotypes. What my group has been trying to do is to study these disorders by generating mouse models that mimic different aspects of the human ciliopathy phenotypes. This includes mice that develop situs inversus. The body plan is reversed, shown here by the stomach on one side of an embryo and on the other side of another embryo, along with skeletal abnormalities, neural tube closure defects, cystic kidneys, hydrocephalus, as well as metabolic disease associated with loss of cilia on a group of neurons in the brain that leads to obesity and inability to control feeding behavior. So what my group has been interested in is trying to understand why does cilia dysfunction cause these phenotypes. We are very interested in understanding what proteins go into making the cilia, how are they assembled and how they're maintained. How is one cilia different from another cilia? In other words, does a cilia in a neuron have different proteins in it than a cilia in the kidney, for example? As part of that, we would like to know how proteins get targeted to the cilia or trafficked into the cilia, how these proteins that are in the cilia function in cilia-mediated sensory and signaling activities, and how disruption of these ciliary functions, these proteins, cause the disease phenotypes that we see in humans and in our mouse models that we generate. We're very interested in understanding the impact that patient variants have on these proteins, and then how this also contributes to cilia dysfunction that causes these phenotypes. With regards to cilia specialization, the way we're approaching that is we have targeted a protein in both cells and mice to a region at the base of the cilia called the transition zone. This enzyme is called BurA, and what BurA does is it adds a biotin to every protein that is transiting into the cilium or out of the cilium. We can then isolate those proteins and conduct mass spec analysis in a tissue or cell type specific way. We can then determine what is the cilia protein composition across different tissues. One of the major interests of my group is trying to understand how cilia dysfunction contributes to cystic kidney disease. Cystic kidney disease, or ADPKD, is a relatively common disorder affecting about 1 in 500 individuals. It's caused by mutations in two proteins called polycystin 1 and polycystin 2 that localize as a complex in the cilia and mediate calcium entry into the cilium. We are very interested in understanding how the polycystin proteins get localized or retained in the cilia, how they're trafficked and processed, their role possibly as a mechanosensor detecting fluid flow over the tubuloepithelium, how patient variants alter this localization, their complex formation channel activity, as well as identifying different pathways downstream of the polycystins that may be druggable to slow the rate of cyst formation. One of the approaches my group is using to understand cilia function in the kidney is called intravital microscopy, or live cell, live animal imaging. And what we have done is inserted an optical imaging chamber into the side of the animal that allows us to look into the kidney, into the tubules and nephrons within the kidney in live animals. And so we can put this mouse onto the microscope and look at changes in ciliary response under different conditions. What we have done is to genetically engineer a mouse that has the cilia labeled with a green fluorescent protein. And what you can see in the normal condition, the cilia are all deflected in the direction of flow. In contrast, if you were to change heart rate or blood pressure, you would see that these cilia respond very differently. These are primary cilia, so immotile forms of the cilia. They respond passively to changes in the flow that's going through these tubules. And this was one of the pieces of the data that led us to suspect that the cilia may function as a mechanosensor that detects that flow going through the tubules. And so we're trying to understand what's downstream, and we note that the polycystin 1 and polycystin proteins are thought to be calcium channels and that they localize in the cilia. So we have built a series of bioreporters to measure calcium changes, and so the level of fluorescence that you see in these cells is relative to the changes in the calcium that occur. We have done this in the cytosol as well as in the cilia specifically, and what you will see is that you do see transient calcium increases in the cilia. You most definitely see changes in calcium in the cytosol. The question now becomes, are these dependent on the presence or function of the polycystin protein? With regards to protein trafficking and localization, the approach that we're taking here for polycystin 2 and polycystin 1 is to tag these proteins, the endogenous proteins, with halo. This is a sequence that will bind to a membrane-permeable fluorescent ligand, and so it allows you to track the proteins in live cells as well as in live animals. Here's an example shown for polycystin 2 tagged with halo in cell lines. And what you'll see is the protein localizes to small puncta within the cell, which we're trying to identify right now what those puncta are, but also prominently in the cilia. And so the nice thing about this is in live cells, you can analyze protein turnover, you can analyze protein trafficking over time. We also want to use these cells for CRISPR-Cas9 engineering to identify genes that are involved in this localization and transport, and eventually using mass spec type approaches, analyze the protein complexes that form with the polycystin 1 and polycystin 2 and determine whether the patient variants alter any of these properties of the polycystin proteins. If you were to summarize my research program, we are very interested in the patient, starting with a candidate ciliopathy disorder. We identify the variants in that patient. We then do an informatic assessment to determine whether that variant is likely pathogenic and the likelihood that the gene impacted by that variant encodes a cilia-associated protein. We test pathogenicity by expressing the variant and the control in xenopus embryos and determine the impact that it has on xenopus development. Shown here is an example for MAPK8IP3, where we express the wild type, versus the R527C variant. You can see a dramatic impact of the variant on development of the xenopus embryo, suggesting that this is indeed a pathogenic variant in that gene. We test whether the gene is associated with cilia by knocking it out in zebrafish through CRISPR-Cas9 engineering. This is an example for CEP290, where you see severe scoliosis as well as development of cysts in the kidney. These are very indicative of a cilia-related gene. We then identify proteins that form complexes with this protein. We determine whether that other members of that complex localize to the cilia. Once we get that information, we then engineer mouse models, and this is an example for B9D1, where we disrupted the gene and show severe craniofacial abnormalities as well as cystic kidney disease. We are really interested in exploring this also in a human relevant cell type. So starting with patient iPS cells or engineered iPS cells, we then go on to make organoids to study disease mechanisms that would be really difficult to study in the context of a human. Our ultimate goal is to identify changes in pathways that are downstream of these genes, hopefully determining whether any of these pathways are druggable that can then go back to improve the lives for the patient with the ciliopathy disorder. So with that, I want to thank you for your interest in our research. I encourage you to please reach out if you'd like to know more about what my lab is involved with, and possibly explore how you could fit into our research program. The contact information is shown on the right, and I hope to hear from you. Thank you.